Welcome Science 10 students to a video on transcribing and translating DNA into polypeptides. This is part of the Google Classroom assignment in which we are working on this transcription and translation practice worksheet, taking a DNA strand and transcribing and translating it into a polypeptide or a string of amino acids. So we are going to be using these two graphics in order to complete this assignment, and you will be using these graphics as well. And so this is a table right here of all the amino acids. Now there's only 20 amino acid types, and these are what form polypeptide chains and then form proteins from that. So as we've discussed, I'll zoom in a little bit here for a sec. As we have discussed with this way of showing molecules, where it just kind of sticks with some. So we have a few different lines with some atoms attached to them. Well, this is an organic molecule in which at each intersection of these lines, you can find a carbon, right? We just don't write the carbons. The carbons help us make our organic molecules. And so these are all the different types, and they will bind together in order to create different proteins. And so they all have different names, and so it's now a matter of we're going to practice taking codons from DNA and turning that into, translating it into these different polypeptides. And so the tool in which we're going to use here is this wheel, this graphic that is showing that it's going to help you map codons to specific amino acids. So one thing to notice with this graphic is that you work from the in and go outwards. So you start from the middle and you go outwards, and noticing that there's these around the outside, there are 3-letter codes, and they correspond to the amino acids, noticing that the amino acids also have these 3-letter codes up here. And so we're going to use this graphic in order to decide which amino acids would go in the chain. So to start, in order for a ribosome to begin reading codons from an mRNA strand and creating a polypeptide, it needs to start with a specific start codon. And so the start codon is labeled on this graphic over here and is methionine. So if we look up in the table, methionine is right here. So MET is the start. So every gene that we're dealing with that codes to a specific polypeptide, which then turns into a protein, each one of those genes is going to start with methionine, and so the ribosome will not start creating polypeptide until this codon is found. And then after that, there's going to be a combination of all these different amino acids in order to create whatever specific polypeptide is required. Okay, so starting with methionine and then move from there. And so in order to read this, you read from the inside out. So the 3-base codon that translates to methionine starts with, remember, from the middle, starts with A, then we move outwards, goes to U, and then to G. Okay. Next, and so if there is AUG, recognizing that we're using U instead of T, so this is reading off of the mRNA. So AUG, if the ribosome recognizes that, or if the transfer RNA that the ribosome allows to attach to this mRNA recognizes it, the creation of the polypeptide will start. All right, so looking at this first example down here, so we have a DNA strand, and the first step is to take the DNA strand and turn it into mRNA. So this is one side of the DNA strand to start, and inside helped by the RNA polymerase, these bases would then attach onto this strand of the DNA. Okay, so that's why we are showing the the opposite or the base pair to the DNA strand. So the cytosine is going to attach to the guanine, and then adenine to thymine, and then uracil rather than thymine is going to attach to adenine. Okay, so this will be in the questions down here. You are doing this part, right? You're taking the DNA strand and you're turning it into the mRNA. Well, up here in this first example is done for you. And so noticing that the beginning is not AUG, right? So this is not going to, this first part here of the mRNA is not going to code to methionine in order to start the creation of a polypeptide. So notice that it's a little bit shifted over. So the ribosome will find this location where AUG exists, that codon, and now it's going to start creating the polypeptide. And so it's going to start reading these codons. And so in the next part here, what has been done is taking that, so the first one, this C base, is not included in this polypeptide, so it can just be ignored at this point, and just coding for these different polypeptides, or sorry, different amino acids. Okay, so the first one, AUG, then you can see that, you can split it up into these 3 base sections, and then just separating them by a dash. Then the next thing is to show the anticodons. So notice that these are just the pairs to a codon. Now the anticodon, this is what is going to exist on the bottom of a tRNA that is going to allow a tRNA to attach to this codon. And so, but what you're actually, in order to create our final polypeptide chain, the information from the codons, not the anticodons, is converted into the amino acids using this graphic up here. Okay, so the first one, AUG, is going to allow the transfer RNA with methionine to attach to it. Okay, well, the next one, so remember we're looking at the codons, not the anticodons. So the next codon of interest is CGC. Okay, so we're interested in which one CGC is going to code to. So we go up to our graphic here. We'll do it in a different color. So we start with C, move on to G, and then G. Okay, and then we see ARG is the amino acid that it corresponds to. And so then if we're not sure what ARG actually stands for, we go up to our amino acid table, and we find it in the top here. And this is arginine. And so we are going to include arginine down below. And so this is what it's done for you. It's taken, so it was CGC, it codes into arginine. And then the same process continues, taking, so AUA is the next codon. Go here, start with A, go to U, and then it goes to A. Going up to the, so the specific, the codon that's, or the polypeptide that it, amino acid that it codes to, ILE, we go up to here, and we find ILE. So isoleucine, right here. And so then we go down to the bottom, and it has coded to isoleucine. And to know when to stop. So just like there's a start codon, methionine, there's also a stop codon. And well, actually there's a few different ones that stop, and they're all down here. And so specific codons will code to this stop, where it tells the ribosome to stop creating the polypeptide, and then it completes. So anytime that you see in a chain, you see UAA or UAG or UGA, it will end the production of the polypeptide. Okay, so UAA, UAG, UGA, and so you can see in this case it ends with UAA. So once it sees UAA, it stops. So noticing that UAA is here in the mRNA strand, and so we don't worry about G, right? And we don't really worry about the C at the beginning. So we have our start codon, we have our stop codon, and everything in between. Okay, so it is now your turn to continue using this graphic, and in order to complete this sheet, the anticodons, I think one warning is do not, so you're taking the codon information and going to the amino acids. Do not use the anticodon line in order to code for the amino acid chain. All right. Okay, so if you have any questions, I will be available on Zoom or through email. Okay, good luck, and talk to everybody later on in class. So have a look at the Moodle page to see what time the Zoom meeting will be. Okay, talk to